now we go to the specifics. The gross features have finished as far as this lecture is concerned. Now we go on to define the nephron and its ultrastructure. Just for revision, these are the two types of nephron, the more abundant cortical nephron and the very functionally important juxtamedullary nephron. So again, an anatomical classification is renal of the nephron is renal corpuscle and long tubule. So as far as its terms are concerned, this whole thing here, this whole thing, this is called the renal corpuscle, which then becomes continuous with the long tubule of the nephron. Goes all the way, collecting duct, and goes into the papilla, and then becomes continuous with the renal pelvis and the ureter, and so on. So renal corpuscle basically includes glomerulus, as I mentioned right here. Okay, mesengium, which is the spaces within the tuft of the capillaries of the glomerulus. It's occupied by cells, and the Bowman caps the space. So this is Bowman capsule, this right here, in which comes in close proximity to the glomerulus, and there is of course a space between glomerulus and the Bowman space. Can't be shown here; it will be shown later. It's called the Bowman space. Okay, so this whole thing is referred to as a renal corpuscle. Okay, and then you have the long tubule. So these two things basically make your nephron. Now, this is important in the sense that it comes in your university question, as a university question. How is glomerulus different from the other capillaries? So we'll talk about that. But first, let's just look at the structure. This is the approximate diameter, 200 micrometers. It's an invagination of capillaries within the Bowman capsule. Its two ends are afferent arteriole and efferent arteriole, as we've mentioned. Now, there are two types of cells, the structure of which we'll shortly be looking at. One is the podocytes, and the other is mesangial cells. Now, the important anatomical difference is that podocyte is outside the basal lamina of the glomerular capillaries, okay, while the mesangial cells is within the basal lamina and the endothelium of the glomerular capillaries. So mesangial cells is part of the wall of the glomerular capillaries, while podocyte is outside, so stuffed in the tufts of glomerular, the whole glomerulus, while mesangial cell is part of the wall. This has important structural and functional consequences. We'll talk about podocyte then eventually becoming part of the filtration barrier, the membrane itself, while mesangial cells, being part of the capillary, provide support, structural support, nutritional support to the cells, and also have been shown to have actin and myosin filaments. So contraction of the mesangial cells as a consequence for the overall surface area of the glomerulus. Okay, so the membrane, the glomerular membrane, permits free passage for neutral. By neutral, we mean electrically neutral substances for up to 4 nanometers. You would maybe think about why we stressed about the neutral electrical status of these molecules. So I'm sure you have read somewhere that the basal lamina, the basement membrane that is full of proteins. It's made up of proteins. It's a thick layer in tissues, and it's negatively charged. So the glomerulus is no exception. It's also negatively charged in the glomerulus. So if you can imagine, if something were to filter through the glomerular membrane, and if it's negatively charged, like a plasma protein, not only will the big structure of the plasma protein be a hindrance to its passage, but also the negative charge on it, on the plasma protein, will be repelled by the negative charge of the basement membrane. Okay, so these are important differences to note. So neutral substances of 4 nanometer diameter size are most welcome to go through the glomerular membrane. It would normally should exclude more than 8 nanometer diameter molecules. And examples of freely permeable stuff is water, small solutes like sodium, urea, glucose, etc. Now, this is where the university question hides. Okay, how is this glomerular capillary bed unique? So number one point is the way the glomerulus is structured between the two arterioles. The net capillary pressure, the hydrostatic pressure within the glomerulus is higher, is quite high actually, as you can see. It's 55 to 60 millimeter mercury. It's much higher than your average hydrostatic pressure across any other capillary membrane, okay, which is around 35 to 40. So this is a high pressure bed. Okay, this is very important. Why is it important? Because one of the main functions where the whole story starts in the nephron is filtration. And in kidney, it's not simple filtration. Filtration happens across all the capillary bed. So nothing fantastic there. But what is distinguished in capillary of the glomerulus is that it has the capability of ultrafiltration. Ultrafiltration. We'll talk about that. Ultrafiltration requires a higher hydrostatic pressure than your average capillary bed. So this is a very important point to note. Secondly, it's filtration only. So it's basically between two arterioles, if you have noticed. It doesn't have a venous end, quote unquote. Both are afferent and efferent, both are arterioles. And therefore, it is filtration only. There is no reabsorption going on across the capillaries. It's made for filtration because the main function of the kidney is filtration. This again is a very important feature. Then the permeability, it is 100 times more because the pressure is more. The structure of the whole thing is such that under high pressure, lots of stuff is supposed to go through the glomerular membrane into the Bowman space and onto the long tubule to be acted upon by the proximal tubule, the loop of Henle, and so on. So the permeability factor here is much more than your other capillary beds of the body. And then you have the last point is that it has a double membrane system for ultrafiltration. Remember this word, ultrafiltration. It has the capability of ultrafiltration because it provides a double membrane system, while the ordinary capillary networks have a single membrane. If you remember, it's just the endothelium of the capillaries, which is the membrane of the capillary system. Here, you have the endothelium, and you have that podocyte cell, which forms the second membrane outside the endothelium. We'll look at it. So these should be 5. There are 4 here. 5th is the fact that it has afferent and efferent arterioles at both ends and no venous drainage. So these should be 5 unique points which make the capillary bed. And remember, this can be asked in a viva or an SEQ. Okay, so we are now looking at the glomerulus. This is actually a micrograph of how it looks like. So look at how small ball-like, tuft-like scenario picture that it has. Okay, so this is the glomerulus in actual. This is obviously a very magnified view of the photograph, and here is the cut edge of the nephron. So obviously it's not a 3D diagram, and the glomerulus, this being the Bowman space, and the Bowman capsule must be around somewhere. It would be continuous with this tube here, which had to be cut and put aside to show you the glomerulus. So you see the cut portion of the tubule, while you see the whole 3D diagram of the glomerulus. This is what it looks like. And when you slit it like that in the frontal plane, just cut it, slice it out. This is the schematic that you get. Okay, so this is how the glomerulus actually looks like. You vertically sliced it through and through, and what you saw is this. So you see the cut portion of the glomerular tuft right here, okay, contained within the capsule. This is the Bowman space, and then this space becomes continuous with the tubule called, this portion of the tubule is called the proximal tubule, okay. Uh, you can see that afferent arteriole is coming in like this. It then forms this tuft, then gets drained into the efferent arteriole, and that's that. I will come to this point at the last. If you notice, there are two types of color within the glomerular tuft. One is this green stuff. You can notice it's really abutting the glomerular capillaries. And then you have this golden yellowish big cell being depicted here and then here. Okay. Now, basically, this cell here is the mesangial cell. And you can see that it abuts, it really is very, very close to the capillary. Indeed, it's part of the structure of the capillary that I just mentioned. Then the green stuff, if you can appreciate, you can actually see the nucleus of this cell and look at the cell body of the cell, okay, right here. Then you have another one here. Then you have another one here. This is, let me just show you this. Concentrate on this one. It's a big cell with its nucleus. And look what it's doing. It's, it's surrounding, it's surrounding the whole glomerulus. It's like it's dominating the whole thing here. And it's giving off these projections all around the glomerulus. And podocyte is that cell. So it basically has a cell body and nucleus residing somewhere, but its projections are far-reaching and as far as it can possibly go. And it's like an octopus. So the octopus has a body, and then the projections of its tentacles that go. That's a good example, actually. So it's like an octopus, which sits here, but its tentacles are all over the place. So they literally surround the glomerulus in a tubular fashion. And we'll talk about how important this is. This is actually the second membrane that forms around the glomerulus. Once we see the ultra high magnification of this area, you'll complete your concept about the podocyte as the second membrane. So finally, we talk about what's happening here. So remember the tubule, which is formed in continuation of the Bowman space. Well, when it becomes the proximal tubule, it then goes on to become the loop of Henle eventually. And as you know, the loop of Henle, let me just go back to show you. Yeah, this is it. So this is where we are right now in ultra magnification. This is the Bowman capsule, which is in continuation with this loop, which is proximal tubule, which then becomes the loop of Henle. And then look closely what happens. It loops back, becomes the distal convoluted tubule here. And then very interestingly, the distal convoluted tubule sort of loops and becomes very close to the glomerulus and then whisks away. It's this area, right, where my cursor is. It's this area which is extremely important physiologically speaking. We'll talk about that. But if you cut it like this in the frontal section, this tube here will look like a roundish structure, right? Because it's a frontal cut. This is what you see. So this is that distal convoluted tubule coming in close touch with the glomerulus, and then it would loop away and form the collecting duct, right? I hope you got that. Now, look closely. When this cut distal tubule, the cells of it facing the afferent arteriole, some interesting stuff becomes apparent. So if you can look close, I don't know if you can appreciate this. You can always open any book, open up Guyton and look at the diagram. Probably it's from Guyton. Generally, the cells are cuboidal, low cuboidal cells, okay, of this distal tubule. But the cells that are facing the efferent arterioles are tall columnar. I hope you notice it. And while it stains darkly anyway, but the tall columnar cells are especially dark. They're very darkly stained. And they indeed, the ones that face the afferent arteriole, they form a disc. And when you look, if you are sitting inside the lumen of the distal tubule and you're looking around in a 3D form, if you look at the other cells, the cuboidal cells, you won't find any especially dark coloration. However, as soon as you put your eyes on the cells which are facing the afferent arterioles, these darkly stained, very darkly stained cells, you would indeed see a disc, almost disc-like structure, because of the very dark stain that these cells take up. And that's why they are called macula densa. Densa means dark, macula means a disc. So basically, it's not a physical disc, but it's just the cells that are facing the afferent arteriole, and they appear like a disc under the microscope, and they stain very darkly. We call them macula densa. Now, this is what the tubule looks like, and the opposing afferent arteriole. Remember, and now the afferent arteriole, as any arteriole, is made up of endothelial cells, basement membrane, and then uh, the smooth muscles that surround the arteriole. Now, very interestingly, the smooth muscles that come across the macula densa, they change, and here they are labeled as blue to show you the difference from the average routine smooth cells. So these are those smooth cells which are not part of anything close to the macula densa. However, these cells which come across the macula densa, they stain differently and they are different. Okay, their function is different. Their function is not primarily contraction, which is the function of the smooth muscle of the arteriole. These cells have a name. They are called JG cells, juxtaglomerular, next to glomerulus, juxtaglomerular cells. So, juxtaglomerular cells of the afferent arteriole are placed opposite the macula densa of the distal tubule, right, of the nephron. Together, these JG cells and macula densa, they form the juxtaglomerular apparatus. Juxtaglomerular apparatus. Okay, and very briefly, a signal from the tubule is sensed by macula densa, which then triggers a response in the closely lying JG cells. And then JG cells respond by releasing a hormone, which I mentioned earlier, renin, a hormone called renin, which then gets secreted inside the blood coming into the afferent arteriole. So signal sensed by macula densa then stimulating or giving that signal to JG cells. JG cell has preformed renin. It can form more renin. It has all the machinery for that. And then renin starts getting secreted more. There's a basal secretion rate. It increases when this signal emanates from the distal convoluted tubule. Okay, this structure is very important, the juxtaglomerular apparatus, and its renin secretion forms one of the mainstays of the long-term uh, regulation of blood pressure. We'll complete this picture as we go along this lecture series. Not this particular lecture, but lectures later on. Okay, so this is the ultra microscopic structure of the whole system. I want you to look at this first. See how the cut capillaries of the cross-sectional glomerulus appear as just cut up tubes, right? I hope you get the orientation. It's just a magnification of any part of this area here. Okay. And you can now appreciate in proper detail the mesangial cell being part of the basement membrane inside, literally inside the wall of the capillary. So this is a rather uh, medium-sized cell within the basement membrane of the capillary. So if this changes its size by contraction or expansion, it will naturally have an effect on the capillary. One of the postulated uh, functions of this cell, and support being one of the obvious functions of the cell. Okay. Now look at Mr. Octopus here. Look at this cell. This is the podocyte. So this is the cell body, the nucleus, and it's a busy bee, as you can see. It has its agents, its processes all around wherever it can get its tentacles on. Okay, so it really extends its processes to far off areas and surround them like this. So this is that projection of the podocyte. Now we are magnifying this bit. Okay, this bit here. How the process of the podocyte, when it comes near the capillary, the glomerular capillary, what does it do? You see that it then divides into many finger-like projections. These finger-like projections are called foot processes. Foot processes. And these foot processes, they wrap around. Now, if I were in physical in front of you, I would wrap around my fingers around a tube or a pencil, and the fingers would be the foot processes, and the space between the fingers would be the filtration slits, as we'll see just in a bit. But let's stay with this diagram first. So this is the foot process. This is, I beg your pardon, the projection of the podocyte, one of several that it does. And here it features when it approaches a glomerular capillary, it then divides into many finger-like projections which wrap around the glomerular capillary segment where it's close to, and makes this whole wrapping of these finger-like projections make a situation where between the finger-like projections, the foot processes, there are slits through which stuff can come out. So the plot thickens. Now, this portion we have further magnified here. So now you can see the proper structure of the filtration membrane, as it's called. So from within the glomerulus, this is within the glomerulus, you see the endothelial cells cut up, and there are spaces between the endothelial cells, which in this case are quite broad. This is another microscopic feature which is distinguished from the other capillary networks. In the other capillary networks, the interendothelial cell space is not this big, but since it's 100 times more permeable, you see that the space is quite big by capillary standards. It's actually called fenestrations, the plural of it. This is that basement membrane, the basement lamina, big lamina, negatively charged, repelling the negatively charged big ions, molecules. Okay. And then now here you see the cross section foot processes that got cut up because we opened it up for you. Okay. And these are the finger-like projections which wrap around the glomerulus. Okay, so very clear here. And within, in between the foot processes, you see a space. This is the filtration slit. This is the filtration slit. So if you were a molecule, you would have to negotiate three things. If you were a molecule inside the glomerulus and you came along on a journey, on an adventure inside the blood from the efferent arteriole, and now you want to see what's going on inside the tubule of the kidney. Sorry. What would you do? You will have to go through the fenestration. You will have to deal with the basement membrane. Then you have to find your way, not colliding into the food process, but rather finding a way in between the food processes through the filtration slit and on to the Bowman space, then the Bowman capsule, and then the tubule, and your journey then takes you to the proximal convoluted tubule, and so on. Here, a point of importance is that fenestration has a diameter of 70 to 90. However, the filtration slit has a diameter of 25 nanometers. Dwell on this for a bit. See what the math tells you. So the fenestration are quite permeable. This is what gives the whole thing a lot of permeability. However, it's not very selective because look at the size. So where does remember, our optimal size is a neutrally charged 4 nanometer particle. It's nowhere near this, right? But look at the filtration slits then. It's the filtration slits which bestow the selectivity of filtration. So through this system, while this is more porous, this here is not very porous. So something which has to go across the filtration membrane, that will have to negotiate with the combined selectivity of the narrow diameter, which is given by the filtration slits. Anyway, so this is what you need to know about the ultrastructure of the filtration, glomerular filtration barrier. And if you want to really complete the picture here, look at this. So you have the capillary, the endothelium, the basement membrane, the cut portions of the food processes of the podocyte. And check this out. The filtration slit actually has a slit diaphragm. So if you really want to complete this whole picture in detail, even the filtration slit has a diaphragm, the slit diaphragm. And there is a hole in between inside the slit diaphragm. And it's not the whole passage which is available for you to go through. It's that hole in the slit diaphragm that the particle needs to go through. Okay, so this is that complete thing. If unfortunately you have to make this diagram, I say unfortunately because it's not an anatomy course. It's physiology. So stuff should be asked which is conceptual and so on. But however, sometimes the examiner is interested to know how do you conceptualize this very important filtration barrier. And for that, he or she may ask you to draw. I think this is a very good diagram to draw. Okay, so make a note of this. The other diagrams are just too detailed and there is limited time in your professional exams. So I think this is a nice schematic diagram which can be drawn quickly. And remember to label it if asked. This is an actual micrograph of the stuff. Look at this. This is, guess what it is? This is the podocyte, this big cell body, and then the processes of the podocyte going everywhere. Look at this. Look at how octopus-like it is, sending off its tentacles, its what you call the processes, and then them wrapping around this whole glomerulus, glomerular tuft. Okay, this is from glomerulus from the outside, and this is the glomerulus from the inside. So you're looking from the capillary wall of the capillary. You're sitting inside the capillary, and see this mesh, this beautiful mesh. You can see the white cells, white blood cells, some platelets as well. And look at this honeycomb kind of appearance. These are fenestrations from within. So I hope this completes your imaginary picture of the glomerulus.